Hello, everyone. It is me again. For those who have been following along and for those who are just tuning in, I am Gardner Butterworth, the head of sales for CellLink Bioprinting in the US and Americas, Middle East, and for our daughter company, Advanced Biomatrix. And I'm excited to enter into our third session of the day. We are joined by Wendy Strigar, CEO and founder of Good Clean Love and Vaginal Biome Sciences. Jeffrey Brown, science advisor at Peta, and Dr. Seyum Ayuni, our own chief scientific officer at MatTech. This trio of speakers will be sharing how they have been working together, modernizing regulatory pathways to improve women's health products. Wendy, Jeff, and Seyum will be happy to answer your questions from the audience. So please make sure to type your questions into the Q&A panel at the right of the screen, and we will answer them at the end of the session. And with that, I'm going to pass the baton over to you, Wendy.
My name is Wendy Strigar, and I am the CEO and founder at Good Clean Love. I am really honored and feel really gifted to be on this path to improve women's health and sexual health products. And at the beginning, it wasn't like this at all. Necessity, in fact, was the mother of invention for Good Clean Love. I started this company because I was trying to save my own sex life and marriage, and it was a true cottage industry. I started this business in my kitchen, so kind of really amazing to think that you could really just be trying to think what you were doing for your own personal needs, and that it could make such a big difference in so many people's lives. At the very beginning, I had no idea I was going to revolutionize or even try to revolutionize women's health and the sexual health care industry. In fact, I really believed that the problems that I was facing were unique to me, that I must be allergic to ingredients in products. But over time, it became clear to me that the kinds of intense reactions that I had to many personal care products, millions of women also experienced. And, as you can see from the icons on the screen, we started out with a lot of idealism. I had this seemingly impossible mission in my business of expanding the experience and awareness of love in the world. Many people told me that was not a mission for a business. And so from the beginning, I was always trying to do things as sustainably as we could. So of course, we were associated with Peta very early on. We were a B Corp as soon as we could be. I'd never considered testing on animals. We were doing the whole organic thing. Everything we could do right was really important to us. To this day, we're the only consumer product company that uses only bioplastics. So, we really were sincere about that. But like everything in the world, we always like to point to one person when we see real change and say, oh, that person did this amazing thing, or we love business success to be attached to one founder, but it's never like that. And in this situation, in terms of modernizing regulatory pathways, I am really honored to be able to present with you with Jeffrey Brown, who is a leader in Peta science and actually kind of was my teacher in helping me sort of understand what was going on and how to work with the FDA. And your own Seyum Ayuni, who has come up with he's the brain trust of MatTech and helped us to prove what we knew was true with his remarkable epivaginal model. So this work would not have been possible without them. So we're going to take turns today, and I'm just going to be kind of an MC as we kind of learn about how we did this. Okay, so there's this standard required animal testing that the FDA has mandated for all personal lubricants. And this didn't really begin until 2013 and 2014, when the FDA really started to enforce this guidance. Previous to that, all lubricants were really treated as cosmetics. And I actually had no idea that I was walking into this FDA operation when I was thinking about lubricants early on. Probably would have scared me away, actually. Even though these mandates are are required of all lubricant companies. Many don't have never done this work. And so there are many, many lubricants on the market that are not 510K cleared. And in fact, in a study in 2018, 96% of consumers reported that they had no idea if their lubricant was cleared by the FDA or not. So, it's not really a public understanding of safety the way that the FDA claims it is, I think. Good Clean Love actually was one of the earliest companies contacted by the FDA. And for us, it was not an option. There's some really interesting information on the internet about, just look up FDA and lubricant, and you'll see that in fact in 2013 that KY was forced to take millions of products off the shelf because they tried to put products on that were not cleared by the FDA. And I just want to say that Johnson & Johnson knew better, but that actually really changed the course of their business in many ways. But this went on for many years. And so what I didn't understand until I was presented with these tests was that I would lose our partnership with Peta by complying with the FDA. And so I was not happy about that. And I wasn't happy to do these animal tests either. So really, that's how I came to know Jeff after we did our first product with all these animal tests. And learned many more things about it, that I decided, okay, there's got to be a better way. So now I'm really proud to produce Jeffrey Brown, who's like a chief at the Peta Science Consortium International and knows more about animal testing than anyone I know. Jeff, it's all you.
Hi, everybody, and thank you for the glowing introduction, Wendy. As Wendy mentioned, my name is Jeff Brown. And I'm an advisor to the Peta Science Consortium International. So if you have any questions about using non-animal methods in place of animal-based tests that are required by any government agency around the world, I'm happy to discuss them with you, so feel free to get in touch. Today, I want to give you a brief summary of how I've worked with Wendy and Sayum to help get Good Clean Love drag the U.S. Food and Drug Administration into the future, and doing that by prioritizing safety tests that reflect human physiology rather than the traditional animal-based toxicity tests that have historically been preferred by the agency. So next slide, please. You may have heard of Peta before, but you may not have heard of the Peta Science Consortium International. The Science Consortium is how we coordinate the scientific expertise of the 23 PhD and master's level scientists who work for Peta entities in the US, in the UK, in Europe, and in India. And when I say that we coordinate scientific expertise, what that means in practice is that we promote the development and the use of animal-free research and test methods. And these are methods that predict human health outcomes better than testing on animals. And we do this in a few ways. Some of those approaches are really relevant to the work that Wendy's doing to modernize the way personal lubricants are assessed. And that includes funding the development and validation of non-animal methods. That includes organizing training opportunities for companies and regulatory agencies who need to become familiar with those methods. And we do that via both in-person workshops and webinars, and also by publishing and presenting our work. And that's especially important where we need to highlight successes and where we need to highlight areas where promising new methods still need a nudge to be widely used and accepted by regulatory agencies. Next slide. It sometimes comes as a shock when people learn just how many products require animal testing to reach the market. And the Science Consortium's work addresses all of these specific toxicological endpoint tests for all regulated products. And that includes personal lubricants and other medical devices. And as Wendy mentioned, in the US, personal lubricants are required to be run through a number of tests that are carried out using animals before they can be cleared for the market. And these are tests that are intended to evaluate the risk of human end users experiencing acute systemic toxicity, skin sensitization, and vaginal irritation after using the product. These animal tests, though, they aren't the only approaches to assessing the risk of these adverse outcomes. So I think an important question to ask is, why are these specific tests that use animals required in the first place? Next slide, Lindy. In the most straightforward sense, these animal tests are required because they've been integrated into the US Food and Drug Administration's guidance. So they've become, in essence, a prescriptive requirement that the agency must promote when product sponsors ask them what tests the agency prefers. And once this has happened, it's very difficult to change that guidance, even as science advances and new test methods are developed that are more relevant to human biology. So it's not quite set in stone at this stage, but it does tie the agency's hands in many ways. In particular, it means that if you ask the agency what tests they require, they'll tell you whatever is written in that guidance. But nevertheless, even as the official guidance documents are very slow to modernize, regulatory agencies like the FDA's Center for Devices and Radiological Health have rules in place that allow and even encourage companies to help the agency understand and accept modern test methods. So in Wendy's situation, for example, the letter of the law that's on this slide, it leaves the door open, and it does that by simply stating that it's the duty of the product sponsor to make and support their claims that the product is safe. So do you see what's missing there? And this is directly from the Code of Federal Regulations. There's no mention of a specific test there. There's only an indication that the sponsor has a duty to support their assertion that their product is safe. And I know that seems subtle, but even though the agency will tell you to use animal tests, if you ask them, how do I show that a personal lubricant is safe? The same agency will open their doors to you if you tell them that I know a better way to evaluate human safety than using the animal methods I've read about in your guidance. And working with Wendy, that's what we've been able to do. The agency is bound by rules of how they are allowed to engage with companies. So asking for permission is a guaranteed way to be told to perform animal tests, and asserting that your understanding of modern science is better than the agency's is how the agency is allowed to open up their process to consider new methods. And in this situation, the FDA has accepted data from human volunteer skin patch testing in place of data from both the rabbit and guinea pig tests that Wendy mentioned. Mm-hmm. And we're in the process of discussing a non-animal approach to replace the use of animals in that one remaining animal test for acute systemic toxicity testing. And I would add that this should come as no surprise. And even though it's difficult for regulatory agencies to make changes to their formal guidance documents, those are the documents that instruct industries on which tests the agency requires or recommends or prefers. Those same agencies have plenty of avenues that sponsors can pursue to make use of test methods that they know provide more useful data when making safety assessments. And none of these pathways is a direct path to a yes from the agency. But nevertheless, there they are. So the FDA's Q submission and pre-submission meeting processes, for example, these have been incredibly useful avenues for Wendy to get FDA's approval to use this human patch testing approach in place of those tests that use rabbits and guinea pigs. And the FDA also has programs like the Medical Device Development Tools Program. And this is intended to be an avenue for groups of companies to be able to join together and collectively get the agency to participate in the process of using new non-animal methods. And this last document that is on this slide, I think is important for everyone to see, because the FDA itself as an agency is in favor of making this transition toward using modern human relevant testing approaches. So this is one of several policy documents that the FDA has produced, and it functions as a roadmap It instructs the public and companies on how the agency is advancing new non-animal testing methodologies. And it also sort of provides instructions on how stakeholders can engage with the agency to bring these modern approaches to the forefront of the agency's decision-making process. So I've got one more slide, Wendy, if you'll move to it. And I think this is a good place to hand it over to Seyum and Wendy to keep talking about this, because what's clear is that the takeaway message is that while regulatory agencies are slow to change, very slow to change, joint efforts by the companies and the researchers are what regulatory agencies around the world, not just in the US, that's what they need to move testing into the 21st century. So it takes a village for sure, but motivated companies like Wendy's are how we move from frustration with the status quo to having breakthroughs that protect human health while leaving archaic animal-based methods in the history books. So I think that's probably a good point to hand it back to Wendy and Seyum.
Thank you so much, Jeff. And I so appreciate your optimism about the FDA. It actually rubs off on me, but I have to say that it does take a village, and also I think really important to not be afraid to be on the FDA radar, right? Because I promise you, I am really on the FDA's radar, in ways that, had I not done this, might not be true. I think systems do want to change and modernize, and the people in them don't always want to do it. So, it takes a while, and it takes, I think, a lot of persistence. But in my case, it wasn't just the animal testing that really forced me to want to make these changes. It was also meeting Dr. Richard Cohn, who pioneered much of the early research in vaginal and rectal biome health. He was a patient science teacher, mentor, and friend that I had prayed to meet for so many early years when I was trying to figure out how to improve the products that made me react so badly. His research and insights literally turned the light on for me. Not only was animal testing morally wrong, but in the case of women's health and sexual health products, it allowed products that were toxic to human vaginas to be continuously cleared. He was one of the lead researchers in the NIH-funded study with the goal of developing a buffer gel to prevent the transmission of AIDS and STDs. Although the gel was not successful, the research with 5,000 women illuminated the important relationship between osmolality and the health of the vaginal biome. For the first time, I understood that my own historic problems were not personal allergies, but the impact of hyperosmolar products on my own vaginal biome. This graph came from the Chemical Engineering News that reported the results of the study that introduced me to Dr. Cohn. This was before I understood osmolality and how critical it was to lubricant safety. But Good Clean Love Personal Lubricant was found to be among the safest products in the study. I cold called everyone quoted in the article to try to understand what this all meant, but Richard was the only one that called me back. Coming to understand osmolality gave me a deep insight into the most common of all sexual health problems and into my own. This graphic shows the gravity of the problem. You can see that when products are up to 30 times heavier, hyperosmolar, they completely strip the vaginal epithelium. And hyperosmolar lubricants are correlated to increase the risk of increased bacterial vaginosis by up to 13%. And 2 in 5 women have BV at any given time. 84% of the time they don't know they have it. And when you have BV and are unaware of it, you're 60% more susceptible to STDs and HIV and 3 times as likely to transmit them. So this actually really galvanized me and made me feel even more fiercely committed to making changes in the procedures that allow personal lubricants into the market. I'm actually still quite shocked at how misunderstood BV is. It's responsible for so many adverse conditions, and still there's so much that we have not understood about the vaginal biome. I liken it to the early days of discovering the power of probiotics for the gut biome. It took a decade for both consumers and physicians to understand that antibiotics were not the solution for gut and overall immune health. We are in the early years of understanding that the vaginal biome deserves the same kind of treatment as the gut biome. And you can see from this slide just how far-reaching the consequences of BV actually are for many women's health. So with all that, I'm super excited to introduce Seyum to talk about the scientific validation of all these things we know with his epivaginal model. It's been a total pleasure to know him and work with him. And he's one of the smartest people I know. So I always found myself in these big meetings with the FDA people, with really smart people, and I had the least to say. So Seyum, with that, please take it away and explain how you learned all these things.
Hi, everybody. Thank you for attending this session, and thank you, Wendy, for the nice introduction. So the market approval for feminine care products from FDA requires basically a battery of assay methods that are biocompatibility. So they use, as the previous speaker said, they use rabbit vaginal irritation assays. They use in vitro skin sensitization assays, and they use in vivo acute systemic tests. And most of them are done in mice, rabbits, and also in guinea pigs. So in fact, the FDA's predictivity of toxicological roadmap calls for optimization of non-animal test methods for safety evaluation of ingredients, drugs, cosmetic cosmetic products, and medical device, and encouraging the shifting in away from traditional animal tests. Next slide, please. So why do we need in the first place 3D tissue models? So the reconstructed tissue model that I'm going to show you in a minute is basically utilize human primary cells. And we use those cells to reconstruct 3D tissue models that's well differentiated and stratified to organ-like structures, which provides greater predictability of toxicity and efficacy of test materials in humans. It is reproducible, and engineered tissues can be produced at higher quantity, which is key for high throughput screening of formulations. Using 3D models for toxicological studies is more translational in overcoming species differences that impede interpretability of preclinical results. So next slide, please. So this slide will walk you through how we just reconstruct these tissue models. So our tissue model is grown on a microporous membrane with or without fibroblasts. And that means we can model different tissues. We can increase different tissue layers and increase complexity of tissues. And in some cases, we have also included immune cells into the system. So what it means is in a more complex tissue model, what we can simulate a heterosexual transmission of HIV and other pathogens such as herpes simplex virus infections. Next slide, please. So this is the structure of the epivaginal tissue that we develop in the epivaginal tissue. As you can see in A, that is the epithelial layer only with the different the tissue layers. It has a apical layer, glycogen filled layer, superbasal layer and basal layers, and it's all grown on a microporous membrane. It's well differentiated and stratified. And in the next, in B, you can see the full thickness tissue model that contains fibroblasts in a collagen gel matrix, like the dermal component of the skin. What in this case, you can see also well differentiated tissue structure. And in C, you can see how the human vaginal tissue model looks like. And our model is basically similar to, it's very comparable to the explant tissue model. That makes it, the structure is similar, and which reflects to the functionality of our tissue models that I'm going to show you for certain materials over here. Next slide, please. So why do we go to the human tissues? Because there are some differences in, this is, for example, a vaginal tissue from rabbits. And the rabbit vaginal tissue is structurally different from that of the human tissue. It only third of the vaginal epithelium is lined by stratified squamous epithelium, and it's unresponsive to most of human genital pathogens. So the results of, for example, the results of the known irritant non-noxinol 9 was variable in this animal model, while we were able to detect it in all testing conditions that it is an irritant. And that this non-noxinol 9 was once tried to be used as microbicide, and it has been tried in humans as anti-HIV agent. And what happens was, because it's an irritant, that resulted in more infection in those people who have been treated with non-noxinol 9. So that has been because of that, it has been abandoned from the test. So what it means is in vitro type of tissue models that we have developed here using human cells can correctly predict the human outcome. Next slide, please.
Oh, sorry. Here.
So this is one of the applications that we have been using, working with Wendy and Richard Cohn. So in here, there are two types of lubricants over here. As it has been mentioned, most of the widely used vaginal lubricants in the US and Europe are strongly hyperosmolar. That makes tissue lubricants 4 to 30 times the osmolarity of a healthy vaginal fluid. The healthy vaginal fluid has osmolarity of about only 370. So the hyperosmolar formulations caused, cause toxicity of tissues and increase susceptibility to infection such as HIV. So our well-characterized 3D tissue model, the human vaginal epithelial tissue model, demonstrates that vaginal lubricants with osmolarity greater than 4 times that of vaginal fluid markedly decrease barrier integrity, compromise tissue morphology by disrupting the epithelium of the epithelial layer. No epithelial damage was observed in this in a hypo or isoosmolar concentrations with less than 400 milliosmolarity per kilogram body weight. So what it means is our tissue model confirms safety concerns of hyperosmolar lubricants and strongly suggests that the usefulness of reconstructed in vitro tissue models as valuable tool for assessing safety of lubricants and other formulations in the absence of direct clinical tests in humans. Thank you.
And here is the work that we did. Do you want to show this, say anything about this, Seyum? This was the research and the histology. So why don't you explain how histology works?
So this shows us that the hyperosmolar lubricants greater than 2,400 osmolality shows the osmolality shows that they were disrupting the upper layer of the epivaginal tissue, and you can see the basal cell layers were okay, but the one which is glycogen filled layer, the suprabasal layer, was disrupted by hyperosmolar osmolar lubricants, and that contributes to the toxicity. Shows the toxicity of these materials, which is basically in agreement with the definition given by WHO for these hyperosmolar lubricants.
So I know we're running out of time, but I do want to say that there's a international global consultation on personal lubricants. This one happened in 2016. I had no idea they threw parties for this, but Dr. Cohn got to go and present this research. And as a consequence, it was included in this report that was put out by the United Nations, sort of naming all of the dangers of hyperosmolar lubricants. And so this science has been pretty widely available for a significant, I mean, a good 10 years. This was from a WHO guidance like right after the research came out, and they limited it to 1,200 isomoles. I feel like that is still actually too high, because truthfully, you can't be just a little hyperosmolar. You know, as soon as you're hyperosmolar, you're actually acting on the tissue in the same way. But this was the minimum that they set at that time. And then I called Seyum every week for about 3 years, and we finally got this published in Toxicology Reports, and this was in 2018. So science takes a long time, and it takes a long time for people to actually see how the work that is readily available is really impactful and can change people's lives. But we've been just keeping at it. And so what we know for sure is that in order for a vaginal biome to thrive and stay healthy, you know, the pH is extremely acidic, between 3.5 and 4.5. And so, you know, we just pay attention to this, and we pay attention to osmolality, but it's the crossroads of the osmolality and the pH that have to both be true in order for products to do no harm. And so you can see on this great graph of all these people making things, that Good Clean Love kind of has that sweet green spot, which both sort of respects the pH requirements and the isoosmotic quality of the formulation. And the really exciting thing is that we started doing this work with the FDA in 2017. And since that time, we've put out 4 products that have all been cleared with the same testing modalities, with only 1 of 3 of the tests. And now Jeff and I are kind of going after it and trying to see if we can get rid of this last test. But no other companies have been allowed to do this. And they, I mean, I don't know, maybe if they, whatever, I don't know what it would take for the FDA to say, here's this other option that companies have used and you can use this. But that, of course, is our goal, that we really want to change the standard of testing at the FDA so that animal tests, which we know are not providing the data that we need, will stop being used as a standard. And we're way over our time, but you know, when I was first invited, I thought we were all going to get to be together and take a boat together out on the harbor Miami, they asked us to define bioconvergence. And so I included my definition here about applying human ingenuity to create innovative approaches to common diseases, thereby creating optimal conditions for well-being. And I really feel like this work that we did all together really sort of sets the tone of what bioconvergence should mean for all of us. So thank you. Sorry we're late. Thanks for attending.
All right. Thank you, Wendy, Jeff, and Sayum. It's amazing to hear about the incredibly important work that you guys have been performing. And the audience has thought it interesting as well. So I'm just going to launch in here, knowing we have a bit of a time limit, to some of the questions. If it can be shared, did Good Clean Love submit epivaginal-based data to the FDA for regulatory approval of any of their products? And if so, was it to address the cytotoxicity endpoint or the vaginal irritation endpoint in the biocompatibility lists of tests required by the FDA?
So this is kind of a complex response. We actually also did a patch test that responded to a human patch test they allowed us to do that responded to some of this. And the epivaginal model is still actually being looked at. Jeff and Sayum can say more about it as a replacement to both of those tests. So Jeff, can you talk about where that is? And Sayum, maybe you want to mention what we're working on?
Sure. And so, like Wendy said, the shortest path to replacing that, the rabbit and guinea pig, the irritation and sensitization test, was to go ahead and use human volunteer patch testing. So Wendy did submit epivaginal data and asked the FDA to discuss it. Is this okay to use in place of any of these three endpoints: cytotoxicity, irritation, or sensitization? And FDA did accept epivaginal from, I think, that first submission in places of the cytotoxicity test.
And 4 times after also.
Yeah, and right, so that's been really thoroughly established as a straightforward replacement. For replacing the sensitization and irritation endpoint tests, the FDA has said that they want to see more product and company representation. So there's a medical device development tool, an MDDT project that's currently underway that's looking at both of those options. Yeah, Sayum, did you have something to add?
I think that's from my understanding, they accepted for cytotoxicity, and the other one is they want us to go ahead and evaluate it against the rabbits. That's where we are.
Cool. Excellent. So up next, what are the biggest hurdles currently in the space, either with the science of the models or from a regulatory point of view?
I mean, I would just say that I'm not as optimistic as Jeff in general, that, you know, it's actually really hard and takes a lot of time and expense and resource to actually work with the FDA and have them accept different models. You know, so the company has to have way more time in their product sort of launch platform in order to make these kinds of changes. And a lot of companies, I think, aren't willing to do that. And, you know, of course, COVID and the way that's affected the agency is also a big factor in that. But, you know, it's actually just stunning to me that I've actually been able to clear 4 products since we did this work, and no other company has cleared a single product since we did this work. So now I feel like I need to come at it from a whole different standpoint and really get this done, because in the same period of time, dozens more products that we know are hyperosmolar have been cleared by the FDA, and that's just wrong.
And I would just really quickly add that I fully agree with everything that Wendy has said. And in terms of hurdles, I think every country approaches this process differently. So introducing a new method that's better, more predictive of the human physiological response. Some countries make it a much more direct path. So most of the countries in Europe, for instance, the European Union has a directive that says anytime a company or a researcher develops a method that doesn't use animals and can replace a required test that uses animals, it should be used with priority. So some of these processes are more straightforward in Europe compared to the US. So that difference between regions has, I mean, it poses a big part of the hurdles that companies like Wendy's have to overcome.
Yeah, I mean, I would even just say that, you know, I mean, in whatever, it's really hard to change large institutions, really hard, but it is possible. There's methods and you have to take the time and you have to pay the money and it's possible. So that's the moral of the story.
Well, I've got a good lead-in question for you then, Wendy. So in the cosmetics industry, consumers drive a lot of the decisions to not test on animals. Do you feel like the consumers will be key to driving the progress with medical devices such as personal health products?
I mean, I think that consumers drive a lot of what happens in our marketplace by how they spend their money. So there is no question about that. You know, I also think that, you know, we have sort of died of the sort of dedication to convenience and ease. And so that kind of complication has made it, I think, easier for consumers to just do the easy thing and not really sort of do the hard work of trying to evaluate. So, you know, I think that consumers historically have driven a lot of change. And I think that if this became an important issue about animal testing in the consumer landscape, it would for sure drive this faster. You know, the truth is that we did this work years ago, and it's been very, very hard for me to get any kind of public recognition around it. It's just come up mostly because BICO has sort of recently started working with MATAC, and so now it's sort of an interesting story again, but we've been doing it for years and years, and most people don't really think about it or know about it.
And Jeff, do you have anything to add? Yeah.
I mean, I think it's clear that when Wendy says people essentially vote for what kind of products they want with their pocketbooks, if a company like Wendy's puts a label on their product that says we don't test on animals unless we have to, and that we're working towards regulatory change, people want to see this. So I think any enterprising company that wants to sort of capture this market energy of consumers is if you're willing to raise this as a question and sort of rally consumer support behind your interest in you know, protecting human health better by moving away from animal testing, that does provide a lot of energy for regulatory agencies to see that they need to step up and do a better job of accepting these methods that companies and consumers both want to put on the market.
Great. So we have a comment in the Q&A about NAMs for vaginal irritation and how this topic can be brought to the ISO level, and that ISO standards give presumption of conformity to a lot of law in the world. On that note, have any of the speakers got comments or experience on the international differences with accepting non-animal models?
Sure, I can jump in really quickly. The interesting thing is that most countries do not require an animal vaginal irritation assay for personal lubricants. So this is one of those situations where it's really the US that's sort of the stick in the mud
laggard,
for progress. So that makes it much less likely that this would elevate to an ISO standard level. It's not out of the question, and I think having that discussion is something that, you know, absolutely comes up in certain you know, contexts, but mostly what you hear is that companies don't want to enter the US market where this test is a requirement because they know this doesn't provide a marketing edge in any country other than the US. There are some exceptions to that rule. I believe South Korea occasionally requires a vaginal irritation test, but this is probably not going to be resolved at an international level. This is an FDA problem.
You know, thanks, Jeff.
Sam, anything to add there?
I think Jeff is the most appropriate person in terms of with FDA. But you know, the other thing is we can apply. For example, now the epivaginal tissue is accepted on a case-by-case basis. And then the protocols have been submitted since to validate this model since 2016, and still we're in the process of evaluating those protocols. So it is slow, and it needs more, you know, fast, you know, push to do it faster and give. Go ahead.
I was just going to say that part of the problem is that a lot of what moves inside the FDA is driven by companies that are trying to get products through. So scientific groups that are working on, sort of validating things, and the FDA doesn't have to do that work with them. They are required to do this work with me. And so that the work they have to do with me has to happen within certain timeframes. But PETA could be knocking at their door all day long. They can take these things, but they don't have to. And I think that's one thing that really influences sort of the pace of this, is that it's like I'm the least probably brilliant person in the room, and yet I'm the one that they kind of need to respond to. And there was a question that I read about whether whether products that are above 1,200 isomoles, which was the guideline that the WHO put out, can still be sold. And yes, many products are still cleared above that isoosmolality and are on the market above that isoosmolality. So customers and people who are listening to this should be conscious of looking at the ingredient index of the products that they might use and making sure that they're not hyperosmolar. Because honestly, it's better to use nothing than to use a hyperosmolar lube. And that's what Sayum's histology proved, and that we then went on to publish it in the toxicology review. That only took 3 years actually to get that done. Science goes really slow. I just want to say that's one of the big hurdles is ask Sayum. I would call him every week. Is it this week?
Excellent. Well, thank you all so much for the lively discussion. I've got a ton more questions, but the producer's in my ear telling me we need to wrap up here. So I want to extend a really big thank you to Wendy, Jeff, and Sayum. If you would like to hear more about MatTech's 3D tissue models and capabilities, or other technologies from BICO, you can head over to the demo sessions, which are starting in the next few minutes. These can be found on the demos tab on the left of your screen. And with that, thank you all very much. Have a great afternoon.
you.